Hey, listeners of the Bio Report. Before we get started this week, I wanted to tell you about the digital library from Deep Dive. How much time does your team spend looking for research papers? Google, PubMed, social media. There's got to be a better way. You can now search a reference database of 100 million scientific papers and read the full text of 20 million articles, annotate them, and share with colleagues. It's the smarter way to do research. Here's the best part. If you're like me and been frustrated by not being able to access articles you find because they're behind a paywall, I've got good news. With Deep Dive, you get one-stop affordable research. If you're a listener of the Bio Report, you can try the enterprise version of the service for free for 1 month. Go to deepdive.com/podcast and enter the code BioReport. That's Deep Dive, DeepDive.com/podcast, and the code is BioReport, one word, all caps. I'm Daniel Levine, and this is the Bio Report. The efforts to develop drugs for neurologic and psychiatric conditions has been plagued with failure. Herophilus is combining organoids derived from patient stem cells along with machine learning to gain new insight into the biology underlying these diseases and discover and develop more effective drugs. We spoke to Saul Kato, co-founder and CEO of Herophilus, about the drug development challenges for these diseases, how his company is using organoids and machine learning to better understand how to target them, and its growing pipeline of therapeutic candidates. Saul, thanks for joining us.
Great to be here.
We're going to talk about Herophilus, the challenges of developing drugs to treat neurologic conditions, and Herophilus's platform to address those challenges. I think neurologic conditions tend to be an area of some of the hardest targets therapeutically. My sense is this is an area with relatively high failure rates for drug developers. Why is that? Is it a lack of understanding of the complexity of these conditions, a lack of animal models, delivery challenges, or is it something else?
I think the reason that neurology and psychiatry as categories have some of the lowest success rates in drug discovery are primarily for two reasons. These are complex conditions of a very complex organ, not well understood how the brain works, and when things go wrong, why they go wrong. So it's complexity of disease, and secondarily, I think because of that complexity, to develop effective therapies, we might have to take more sophisticated approaches. I think those are the two major issues. The other issues that you brought up, I mean, animal models are certainly more challenged. The translatability of behavior between species is hard, and of course there are challenges of getting into the brain. But to be fair, plenty of drugs do get into the brain.
Herophilus has developed a platform for discovering therapies for neurologic diseases using patient-derived human models known as organoids. What's an organoid, and how do you create them?
Yeah, so an organoid is somewhat kind of a scary science fiction sounding word, but it is the word that the industry and the field has settled on, and it sounds fancy and new, and in ways it is. It's a new way to culture cells, typically stem cells, typically derived or reprogrammed from patient samples, either blood or fibroblasts. But when you culture cells in a traditional flat-bottomed, say, glass dish or well on a well plate, the cells and neurons in particular tend to spread out, and they don't form cohesive tissue. What was discovered, and these sort of discoveries stemmed from some of the pioneering work in the 90s in Japan, primarily by Sasai and Yamanaka and the folks that figured out how to make inducible pluripotent stem cells, iPSCs, which have revolutionized the field of culture, human-derived culture models. They also figured out that if you change certain aspects of the way that you culture cells, some of the sort of milieu, the setting, the physical setting, as well as provision of certain nutrients and molecules, the cells will sort of self-aggregate into tissue-like cultures. And depending on how you steer the fate of these cells, you can get these cultures to appear as if they're forming brain tissue. That would be a brain organoid. Lots of success has been shown to derive other organ types. Virtually every organ in the body now, someone has developed a protocol to produce these organoids for. So it sounds kind of crazy and weird, and I think it should be appreciated that in some ways this is actually a more realistic culture than formerly from a traditional what was called two-dimensional or dissociated culture. In ways, this is just a more natural way to grow stuff.
Do you need to start with cells from a patient with a disease to get an organoid model of that disease, or is this something you're able to engineer?
Yeah, you can start with an iPSC generic line, a healthy line, and use CRISPR or other gene editing technology to engineer models. That's really nice because you generally then have an isogenic control. You have another culture with exactly the same genetic background, except for the edit that you hope that you made. But we tend to start always from patient samples, and the reason for that is even for monogenic disease, but particularly for more complex diseases. We believe that you need to study a distribution of genetic backgrounds to get a realistic picture of the patient population and how the disease arises from their genetics.
How well do they actually replicate neural activity in the brain?
Yeah, that's one of the most profound features of these new culture models, is that they really exhibit both realistic neural activity, spontaneous network activity, neurons electrically discharging, communicating with each other, and producing spatiotemporal patterns of activity that really resemble in vivo recordings. It's quite profound if you're a neuroscientist and you see one of these cultures for the first time, how realistic they look, and they really are a step up from the kind of electrical network activity you can induce in a two-dimensional culture. But in addition to neural activity, you replicate quite a bit of the other sort of large, obvious biological features, such as gene expression and, I think quite profoundly, the cytoarchitecture of tissue. So when you look at these under a microscope, these cultures, they really resemble the emergence, what you see when you watch embryonic emergence of tissue. The neurons order, they layer, they form kind of classical, in the case of cortex, cortical structures that you expect to see in a developing brain.
Well, walk me through the platform. What's the process? How do you take a sample, run experiments, and end up with actionable data at the other end?
So everything starts with a patient sample, in our case. This is, again, blood or fibroblasts from a skin biopsy. Mostly we're starting with patient blood. It's easier to get. We reprogram those following a fairly standardized protocol to generate inducible pluripotent stem cells, iPSCs. And then there's a sequence of culture steps to go from iPSCs to organoids, or to these sort of growing brain cultures. And once we have the brain cultures, we then sort of subject them to a battery of biological assays to read out what we call phenotypic data from these cultures. And this is before doing anything with regard to discovering drugs. We are developing a model of disease. So we profile all of the phenotypic characteristics of organoids that come from patients with a genetic background of disease, and we compare that to healthy controls. Once we've built up that multi-phenotypic profile, which we call a deep phenotype, we then have a set of biological criteria to screen therapeutics against. And then the process starts to resemble sort of things from the current mainstay of drug discovery, high throughput screening, for example. We will generally extract some aspects of the deep phenotype into something that's scalable, what we call a primary assay, and we'll screen against that. And then follow up when we have a set of therapeutic hits with a deeper secondary deep phenotype screen.
How much uniformity or variance do the organoids have, and what's the consequence of that from a drug discovery and development point of view?
Yes, with any new model, there is a process to industrialize or to tame the model. This is true for new protocols in two-dimensional culture, but also particularly so for these three-dimensional or more sophisticated culture methods. And organoids, cerebral organoids appeared really on the scene around mid 2010s, 2013. And initially, there was a lot of concern that variability across many different axes was going to render organoids just kind of too messy for real business. As an example, the gross morphology of organoids, depending on the protocol, can vary quite widely. Starting with the exact same sort of initial conditions, you'll get organoids of wildly different shapes and sizes. They'll possess different mixtures of tissue types and cell types. And that scares any rigorous scientist that wants to develop statistically sound measurements. However, a lot has been figured out in the last 10 years about how to tame the organoid, if you will, or reduce variability. Both work at our company and in many other labs. And the good news is that we have really reduced a lot of the variability across many of the axes that biologists consider important for reading out sort of biologically meaningful signals. And we also have figured out a set of knobs to sort of control the level of variance. And so it turns out that you can grow organoids in many different ways. And these ways live on a spectrum from highly uniform, but maybe stripped down of sort of the diversity of function and of the heterogeneity, for example, the tissue and complexity of the tissue. And you can get, for example, very nice, beautiful spherical organoids if that's important. If you think that's important as the experimenter, or you can turn up the sort of the freedom to self-emerge by playing with some of the protocol parameters, and you can get organoids that sort of develop into multi-brain area organoids, and those will tend to be more variable. And so you can reduce variability through a set of protocol optimizations. That's what's gone on over the last 10 years. And then another way to deal with variability is really using replicates, right? So this is building up statistical strength simply by repeating experiments. That's another big motivator for taking what like many biotechnologies start as artisanal sort of hand crafted work in small labs and scaling it up.
The organoids you develop are part of an automated process that uses machine learning and deep analytics. What kind of data are you able to get on the organoids and how they respond to a drug or act in a disease state?
Yeah, that's one of the beauties of this sort of intact or full systems approach, because the organoids have a rich set of biological features across different modalities. And so we collect all kinds of data. We collect cytoarchitectural microscopy, so pictures of where the cells are. We measure the composition of the cells, the different types of neural subtypes that emerge. We look at gene expression. We look at metabolomics, both in secreted media and intracellular metabolomics. And we also look at functional activity. So we look at the neural activity that emerges in the neural networks that grow. And so you're really getting a full picture of biological processes. And of course, that means you have a lot of high dimensional heterogeneous data. And we use machine learning to sort of distill that down into a classifier or a picture, just a simpler description of a disease state that we can then screen against.
You're developing therapies for both neurodevelopmental diseases and for neurodegenerative diseases. The neurodevelopmental diseases you're pursuing are largely monogenic, although not exclusively. Does this change how you think about how you target the conditions and screen therapies?
Yeah, absolutely. So the complexity, the etiological complexity of diseases, where the root causes really will fall on a spectrum from monogenic disease, where many rare diseases are monogenic in nature. One gene is really known to be the root cause of the disease. And that is very attractive for the drug hunter, because you kind of know that you should probably target either that gene itself or the gene underlying the product that is faulty in a disease state, or maybe one step downstream in a pathway. But you can be kind of focal in your approach. That starts to go away as you get into more complex diseases, where you suspect that more than one gene or gene product, the interaction of gene products is at play in causing a disease. And that's probably the case for more complex diseases in sort of the more diffuse neurodegenerative categories, and also some neurodevelopmental disease. We started with a monogenic disease because of this sort of the feeling that it would be easier to prosecute. But the intact systems nature of our approach is really designed to be able to go after more complex disease. So beyond monogenic disease, for example, we're looking at a disease called 22q11 or DiGeorge syndrome, and that's a result of a large deletion covering about 35 genes and also other intronic regions, but that disease is kind of, we might call it, between monogenic and highly polygenic. And so, we sort of look at the world as kind of climbing up from monogenic disease and getting into polygenic disease.
Your lead indication is in Rett syndrome. What is Rett syndrome, and how does it manifest itself and progress?
So Rett is a really tragic disease that affects 1 in 10,000 female births. I think cumulatively there are about 350,000 in the world Rett patients. It comes on very early in age, as early as a year to a few years old. Are typically first diagnosis is made, and it exhibits a wide variety of symptoms, severe neurological symptoms, as well as other peripheral symptoms. The neurological symptoms include seizures, deteriorating motor function, apnea, and a lot of autistic-like symptoms. And this is a really devastating disease, because it sort of seems to come out of nowhere, much like autism when a girl is very young. It's a monogenic disease. The gene was discovered in 1999, MECP2 by Huda Zogby, and this is provides hope and purchase for drug development programs. We can target the disease at its root, and I think because of the work done there and the knowledge in the disease biology, and because of the severity and the high unmet need, this is a disease that many targeted approaches, new biotechs go after.
You've identified a lead candidate. What do you know at this point about the drug, and where are you in the clinic?
Yeah, so we took a particular strategy. There are a number of strategies to try to rescue or cure this disease. And our particular strategy, I should mention that Rett is, and the reason I only mention females is that it's an X-linked disease. It is typically lethal in boys. This is a haploinsufficient disease. And every neuron, for example, will have either a healthy or an unhealthy copy of this gene. And if they have the unhealthy mutated copy, that cell's not going to produce the gene product. And the gene product of MECP2 is an early transcription factor. So one of the there are different strategies. You could pursue a first generation gene therapy approach and just try to overexpress or express in MECP2. The risk there is that overexpression of this transcription factor produces a disease as severe as Rett, MECP2 duplication syndrome. So we looked at that and we decided to take an approach that others are also pursuing, which is X reactivation. So the idea is that if we can just turn on the healthy silent copy in neurons, then we'll sort of have an automatically dose regulated gene expression approach. And so we conducted a screen for small molecules for X reactivation. And we did find one, and we hope to be in the clinic next year.
Your lead indication in the area of neurodegenerative diseases is in Alzheimer's disease. There's been a lot of debate about the targets that drug developers have used in pursuing Alzheimer's because of the large number of late-stage failures. How well do you understand the target when your platform identifies a potential therapeutic?
Yeah, so another great feature of these intact systems is that we can do target discovery, validation, deconvolution. So you can start with a phenotypic approach where you screen for drugs that rescue the features, microscopic features exhibited by our disease and our disease models, and then go back and try to understand if you find a perturbatant, a pharmacologic perturbatant or a therapeutic that modifies the phenotypes, then we can go back and understand with more clarity how this is actually the therapeutic is working, and thereby discover the targets, the molecular targets involved in the disease. And of course, there's always been a challenge in Alzheimer's around getting confidence around what we call the arrows of causality. There are clear signals of disease in misfolded proteins, in things like A beta 42/40 ratios, and there's been quite a bit of molecular characterization, and there's no doubt that those are concomitant with disease progression. But the risk has always been, and the question has always been, which way is the causality arrow pointing? In other words, are these measurables causing the disease, or are they another side effect of disease progression? And so that's one of the great things about our system is we can really, pause and take a longer time in the preclinical development phase to really understand what is going on in the disease, what the molecular components are, and which are causal to which. And so a great deal of work is being done now to sort of take redoubled efforts to understand these sort of etiological network of Alzheimer's. And we're quite active in that area now.
Well, where are you in this program, and when might you be in the clinic?
Yeah, so the downside of taking a measured approach, of course, is that we are probably quite a ways off from human clinical testing, unfortunately. We do think we should really make sure we understand and have a higher degree of confidence about the biology that we're observing and how this all works. So unfortunately, I don't have a great story for you there.
Saul Kato, co-founder and CEO of Herophilus. Saul, thanks so much for your time today.
Thank you.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.